Imagine holding the complete instruction manual for a human being, a brand new version, written from scratch. Not just a faded copy or a translation, but a pristine, detailed guide that reveals every secret, every function, and every possibility hidden within us. This is no longer science fiction. As of July 22, 2025, it's reality. The world watched as scientists unveiled a milestone that will echo through history, a moment when the impossible became possible. For decades, we read the human genome like a book with missing pages. We could only guess at the words lost to time, piecing together fragments and hoping for clarity. Now, we can not only read the entire story, but also write our own chapters. We're no longer limited to what nature has handed us. We can add, edit, and even create new instructions. This breakthrough is like discovering how to build a pyramid, not just finding one. It's the difference between admiring a masterpiece and learning the artist's every brushstroke. Writing DNA lets us test ideas, build custom tools for our cells, and dream up solutions once thought impossible. We can design new therapies, engineer cells to fight disease, and even imagine life forms that have never existed before. We're moving from passive observers of biology to active participants in our own health and evolution. The power to shape life is now in our hands, and with it comes both incredible promise and profound responsibility. With a complete human genome, we finally have the ultimate guide to tackle challenges like cancer, genetic disorders, and aging. We can identify the roots of disease, predict risks, and personalize treatments like never before. We can see every part, understand connections, and pinpoint problems at their source. The mysteries of our biology are coming into focus, one gene at a time. This isn't just about better treatments, it's about understanding the why behind our biology. It's about unlocking the story of who we are and how we came to be. Thousands of scientists, decades of research, and relentless curiosity brought us here. Their dedication has built the foundation for a new era in science and medicine. We're about to explore what a genome is, how it's built from the ground up, and what this power means for our future. The journey ahead will change not just medicine, but our understanding of life itself. Buckle up. We're diving deep into ourselves to witness how science is rewriting the code of life. The adventure is just beginning, and the discoveries waiting for us are beyond imagination. The future is being written, and we're learning the language. Together, we stand at the threshold of a new chapter in the story of humanity. So, what is a genome? It's a word you might have heard in science class or on the news, but what does it really mean? Think of your body as a complex city, with millions of moving parts, and the genome as its entire library of blueprints, an instruction manual for every building, street, and system. Every single cell in your body contains a copy of this library, written in a unique code made up of just 4 chemical letters, A, T, C, and G. These letters form the language of life. The human genome is massive. It contains about 3 billion of these letters, carefully organized into 23 pairs of chromosomes, each pair holding vital information. You inherit one set of chromosomes from your mother and one from your father, making 46 chromosomes in total. This unique combination is what makes you, you. Each chromosome is a long, coiled strand of DNA, and within these strands are specific sections called genes. Think of genes as recipes in the blueprint, each with a special job. We have about 20,000 genes, and each one contains instructions for building proteins, molecules that determine your traits, from eye color to how your body fights disease. But genes are only part of the story. There's much more hidden in the genome's vast code. The so-called junk DNA, which doesn't code for proteins, is actually crucial. It acts like a control system, deciding when genes turn on or off and how much protein to make. This regulatory DNA orchestrates the entire symphony of life, ensuring every part of your body works in harmony, just like instruments in an orchestra. Without this control, our cells would be chaos, unable to function properly, leading to confusion and disease. Reading the genome sequence is like deciphering the fundamental code that makes you, you. Scientists use powerful machines to read and interpret this code, unlocking secrets about health and identity. Even a single typo, a tiny change in the code, can mean the difference between health and disease, showing just how precise this blueprint must be. That's why having a complete, accurate genome is so critical. It allows doctors and researchers to understand, diagnose, and even prevent genetic disorders. The genome is the master key to understanding how our bodies are built, how they function, and how we might one day fix what goes wrong. In the end, the genome is the ultimate user guide for being human, a living book that tells the story of life itself. How do you write a human genome from scratch? It's a question that pushes the boundaries of science and imagination, blending biology with engineering in ways that were once the stuff of science fiction. Imagine it like assembling a massive encyclopedia, but instead of words, you're piecing together the fundamental letters of life, one by one, with painstaking care and precision. Scientists start by synthesizing the 4 DNA letters, A, T, C, and G, in the lab, using advanced machines called DNA synthesizers. Each letter represents a chemical building block of our genetic code. These machines print out short snippets of DNA, just a few hundred letters long, but with incredible accuracy, ensuring every sequence is correct. Thousands of these tiny snippets are then carefully stitched together, forming longer and longer strands like assembling sentences into chapters. Here's where nature lends a hand. Yeast cells act as microscopic factories, expertly joining DNA fragments into full chromosomes, something machines alone can't yet do. Researchers insert the synthetic DNA into yeast, which assembles the pieces with remarkable skill, almost like a master bookbinder binding pages into a book. This process is repeated over and over, gradually building up entire chromosomes, the long threads that carry our genetic instructions. The ultimate test comes next. Scientists package these synthetic chromosomes into a living cell to see if they can function and support life. In a sense, we're learning how to compile this biological code and run the most complex program ever written, the program of life itself. It's a breathtaking feat of engineering, transforming digital information into the physical blueprint that makes every living thing possible. Piece by piece, step by step, we're learning to master the code of life, unlocking new possibilities for science, medicine, and beyond. The challenge now is to make this synthetic DNA work seamlessly inside living cells to truly bring these blueprints to life. This is the new frontier of synthetic biology, a place where imagination, technology, and biology converge to rewrite what's possible. The Human Genome Project gave us 92% of our genetic sequence, but the last 8% was missing, repetitive, hard-to-read regions. These gaps were in crucial areas like centromeres and telomeres, essential for cell division and chromosome protection. Without them, our map of the genome was incomplete. The Telomere to Telomere (T2T) consortium set out to finish the job, using new sequencing technologies to read long stretches of DNA. In 2022, they succeeded, producing the first truly complete human genome. For the first time, we could read our instruction manual from start to finish. This complete T2T genome is now the gold standard, making synthetic genome construction possible. Before, missing instructions meant incomplete or faulty results. Now with the full sequence, scientists have the exact template they need. This finished map unlocks the next leap, writing a human genome from scratch. Having the complete genome is just the beginning. The real revolution is writing it. With a synthetic genome, we can change or remove parts and see what happens, revealing the function of every piece of DNA. It's like building a custom engine and testing each part to see what it does. This approach will help us map the function of thousands of genes and regulatory elements. We can design minimal genomes with only essential genes or enhanced genomes with new abilities. Imagine cells engineered to produce medicines or resist disease. Biology becomes an engineering discipline. We can design, build, and test new biological functions. This shift from reading to writing DNA accelerates discovery. We're moving from observing nature to creating it. The possibilities for medicine, technology, and understanding life are limitless. Synthetic genomics is transforming how we ask and answer the biggest questions in biology. The future of life is now something we can design. Building a human genome from scratch could revolutionize medicine. Genetic diseases like cystic fibrosis or sickle cell anemia are caused by tiny errors in DNA. With synthetic genomics, we could create healthy, error-free chromosomes to replace faulty ones, offering true cures. Cancer, a disease of the genome, could be tackled by engineering immune cells programmed to target only cancer cells. We could also study cancer by building cells with specific mutations, testing new drugs with precision. Synthetic genomics lets us explore aging, designing genomes with enhanced repair mechanisms or longer telomeres. This could unlock secrets to healthier, longer lives. The journey is just beginning, but the tools are here. We can now build and test bold ideas in the lab, Medicine is shifting from treating symptoms to engineering wellness. Personalized treatments could one day rewrite faulty DNA, restoring health from within. The future of medicine may be written in synthetic DNA. The promise of synthetic genomes is huge, but the challenges are just as great. Writing 3 billion DNA letters with perfect accuracy is slow, expensive, and error-prone. We need faster, cheaper, and more reliable DNA synthesis. Epigenetics adds another layer. Chemical tags on DNA control gene activity, and we're only beginning to understand them. Building a synthetic genome isn't enough. We must also recreate these regulatory marks. Delivering synthetic chromosomes into living cells and making them work is a massive technical hurdle. Swapping out entire chromosomes is beyond current capabilities. Much of the genome remains a mystery. Building on incomplete knowledge is risky. We need more research to understand every gene and connection. Progress will require patience, innovation, and global collaboration. The journey is long, but the potential rewards are extraordinary. Writing a human genome raises profound ethical questions. Safety is paramount. Unintended flaws could create new diseases. Access is another concern. Will only the wealthy benefit, deepening inequality? Where do we draw the line between curing disease and enhancing traits? There's also the risk of misuse, from designer babies to biosecurity threats. Ethics must guide this science with global conversations and strong safeguards. We stand at a turning point, able to read and now write our genetic code. This is a shift from inheriting biology to authoring our destiny. The path ahead is challenging, but the potential to alleviate suffering and expand knowledge is breathtaking. The questions raised belong to all of us, not just scientists. Be curious, ask questions, and support science education. The story of the genome is about human curiosity and our drive to understand life. Every one of us has a role in shaping the future. The creation of a synthetic genome is a powerful tool for a healthier, more resilient world. Let's embrace this opportunity with wonder and responsibility. The next chapter is ours to write. Let's make it a good one.